A very good afternoon to everyone. Welcome in today's class. And as we say in the starting, let's crack NEET PG together. So, the topic of today is carbohydrates. We are going to talk about the basics of carbohydrates in detail. So, the topic of So, let's crack NEET PG together. The topic of today is carbohydrates. We'll be talking about carbohydrates after a small introduction about Unacademy. So Unacademy is one of the India's largest platform for the preparation of NEET PG examination, and India's best educators, they teach at Unacademy. So you can get this benefit of getting mentored by the best faculty members all over India. Then there are special classes, and what is so special about these classes is that you can ask your doubts, queries live with the faculty member, and they'll be very much willing to solve your doubts and queries. Then for qualifying any kind of competitive examination, it is very much important to attend the PYQs and MCQs related to the examination. So that is also possible with Unacademy because there are free mock tests, weekly tests. So you can anytime go and attend these tests and you can become well versed with the pattern of examination. Then how to download the app? Just go on Play Store, write Unacademy, and you will be directed towards the Unacademy learning app. Download the app and make your account, log in, and you are good to go. You will have access to all the free lectures available at the platform. So you can just go through the lectures. And what are the benefits of preparation with the help of online portals such as Unacademy? There is flexibility in the schedule. The language can be of your choice because there are many educators who even teach in the regional languages. Then unlimited practice. Once you have given a quiz, then you can just redo the quiz and rewatch the videos as many number of times as you want to. Then you won't feel anything different than a normal classroom because you can chat with your educator. Then there are interactive polls. You will need to answer some of the questions which are asked by the educators. So interactive polls are there. Then learn anytime, anywhere is the benefit of online learning. Then there is this plus subscription, and what is this plus subscription is that in plus subscription what happens is you will get all the videos of the 19 subjects in the pre-recorded manner, and faculty members they take these short-term courses and crash courses for all the upcoming examinations, so you can very well be updated about the upcoming exams. And then you need to pay a certain amount for this plus subscription, and that is also quite pocket friendly. And if you're going for a 12 monthly or a 24 monthly plan, then it is quite reasonable, and then you can also make the most out of it because there is so much of data available, so many video lectures in a very interesting manner, so you can just watch these videos and make the most out of it. So for doing this, you can just pay a certain amount and get the plus subscription. And in this plus subscription, you will be getting a 10% discount if you are using my referral code, that is FB01. So do not forget to use my referral code, that is FB01. So by using my referral code FB01, you will be getting an additional 10% discount, right? So now moving forward with carbohydrates. Carbohydrates. So they are actually hydrates of carbon. They are actually hydrates of, they are hydrates of carbon. So previously they were called as hydrates of carbon, but now it's a little bit different. So that also will be seen. So hydrates of carbon, what is the formula? It's CN, H2N and ON. So this is the hydrates of carbon, hydrogen and oxygen. So hydrates of carbon. There are some exceptions which actually fall in the category of this formula. So the exceptions for this formula are lactic acid and acetic acid. Lactic acid and acetic acid. So lactic acid and acetic acid, they both fall in the criteria of satisfying this formula, but they are not actually carbohydrates. So they satisfy the above formula, but they are not carbohydrates, right? Then there are some of these sugars who does not satisfy the formula of CnH2nOn, but still they are carbohydrates. So let us see other exceptions in which they are not satisfying the above formula, but they are carbohydrates. So example is deoxyribose, deoxyribose. So it is having one oxygen less. It is having one oxygen less than the ribose. Less than ribose. So it is not actually falling in the formula of CN, H2N and ON, but it is very, very important sugar of DNA. So it is important sugar in DNA. It is important sugar in DNA, and it does not satisfy the above formula, but it's a genuine carbohydrates. It's a genuine carbohydrates, right? So now coming to the definition, that after seeing the exceptions and after seeing the older definition and seeing the formula, let us come to the definition. What is the definition? So the definition, the literal definition goes like this: carbohydrates are polyhydroxy aldehydes and ketones. They are polyhydroxy aldehydes and ketones, or compounds which produce them on hydrolysis. Which produce them on hydrolysis. This is the definition, that carbohydrates are polyhydroxy aldehydes and ketones, or compounds which actually produce them on hydrolysis. This is the literal definition of carbohydrates. Now, talking about the classification of the carbohydrates and seeing their examples. So, classification. Classification and along with the examples. So first classification is monosaccharide. Monosaccharide. So monosaccharides, it means single. Mono means one. So one sugar molecule is called as monosaccharide. It is also called as simple sugar. Simple sugar, and it cannot be hydrolyzed further. Cannot be hydrolyzed further. Cannot be hydrolyzed further, and it is made up of the single unit of sugar, right? So monosaccharide. Then talking about disaccharide. So itself the word is saying di means two. So when the two sugar molecules, they are joined with each other, they are called as disaccharide, and the bond which is connecting them is glycosidic bonds. So two sugar molecules connected by glycosidic linkage. Connected by glycosidic linkage is called as disaccharides. Glycosidic linkage is disaccharides, right? Then moving ahead and talking about the oligosaccharides. Oligosaccharides. So 3 to 10 sugar units or monosaccharide units make oligosaccharides. So 3 to 10 monosaccharides make oligosaccharides. Then polysaccharides, more than 10 sugar molecules. More than 10 sugar molecules make polysaccharides, right? So we have seen what is mono, what is di, what is oligo, and what is poly. Now talking about the individual classes and their example. So first one is monosaccharides. Monosaccharides. So they are classified on the basis of number of carbon atoms. Number of carbon atoms. Like whether it has got 2 carbon atoms, like 2 is not possible, 3 carbon atoms, 4 carbon atoms, 5 carbon atoms, and then they are classified on the basis of functional group. Functional group. So number of carbon atoms and functional group. So the 3 carbon monosaccharide is called as triose. Then 4 carbon monosaccharide is called as tetrose. 5 carbon monosaccharide is called as pentose. 6 carbon monosaccharide is called as hexose. And 7 carbon monosaccharide is called as heptose. So this is on the basis of number of carbon atoms. Now if we are saying that on the basis of functional group, then it is divided on 2 bases, that is aldolase and ketose. Aldolase and ketose. So the sugar having aldehydic group falls under aldolase, and the sugar molecule having the ketone group fall under the ketose. So talking about the first one, that is triose. Triose having 3 carbon as the basic unit. So its aldose form is called as glyceraldehyde. And its ketone sugar is called as dihydroxyacetone. So for triose, the aldehydic form is glyceraldehyde and the ketonic form is called as dihydroxyacetone. Now for tetrose, the aldolase form is called as erythrose and ketose form is called as erythrulose. Then hexose, its aldehydic form is called as glucose and its ketonic form is called as fructose. Then for heptose, the aldehydic form is called as glucoheptose and its ketonic form is called as pseudoheptose. Right? Now, talking about the tests of monosaccharides. tests of monosaccharides. What are the tests of monosaccharides? So for carbohydrates, it's Molish test, then it is Benedict's test, and this Benedict's test is specifically for reducing sugar. Then Barfoot's is for monosaccharide unit. Then Selvinov's is for the ketone sugar or ketose sugar. So it is somewhat MBBS. So the test of monosaccharides can be remembered MBBS: Molish, Benedict's, Barfoot's and Selvinov's. Right? Let us move ahead and let us talk about disaccharides. So the two sugars, when they are joined together by the glycosidic linkage, they are called as disaccharides. This we have already seen. Now these disaccharides, they are of two types: reducing types and non-reducing types. So first of all, talking about the reducing types of disaccharides. So first one is lactose. So lactose, it's a milk sugar. Then it is made up of galactose and glucose. And the linkage which is present in lactose is beta 1,4 glycosidic linkage. Then instead of calling it directly lactose, there is another name also for it, and the name is beta D galactosyl. 1,4 beta D glucose. Then the tests for the sugars for the disaccharides, it is and monosaccharides is the OSA zone test. So whenever the OSA zone is done for lactose, it gives the powder puff or the blooming kind of needles. So for OSA zone, the structure which is obtained for lactose, is powder puff. It is powder puff type of this type of OSA zone we will be getting. This is the OSA zone for lactose. Powder puff, typically called as powder puff OSA zone. Then talking about the second type of disaccharides, that is maltose. So this maltose is actually made up of glucose plus glucose, and it is joined by alpha 1,4 glycosidic linkage. And its another name is alpha D glucosyl 1,4 alpha D glucose. The linkage in maltose is alpha 1,4 glycosidic linkage. And the structure which is obtained for osa zone of maltose is a sunflower kind of structure. Osa zone of maltose gives a look of sunflower shape. Sunflower shape, maltose, right? Now, talking about the non-reducing sugar. Non-reducing sugar, and that is in the category of disaccharides. So, the non-reducing sugar falling in the category of non-reducing sugar is sucrose. And sucrose is made up of glucose plus fructose. Glucose plus fructose. And the linkage is alpha 1 beta 2 glycosidic linkage. Alpha 1 beta 2 glycosidic linkage. And why it is non-reducing sugar? Because the functional groups are attached in the bond formation. So functional groups form the bonds. Form bonds. Thus, they are not free to form the further bond. They are not free for the further reaction. So they do not give osa zone. Do not give osa zone. Because the carbon number 1 of glucose and carbon number 2 of fructose, they participate in the bond formation and they do not give any kind of osa zone. Now, if we are talking about the osa zone of monosaccharides, osa zone of monosaccharides, osa zones of monosaccharides. So the monosaccharides, they give broom shaped osa zone, broom shaped or hay shaped, broom shaped, hay shaped or needle shaped osa zones. The monosaccharides, their osa zone is broom shaped, hay shaped or needle shaped. So we have talked about the monosaccharides and disaccharides. Now talking about the examples of oligosaccharides. Oligosaccharides. So they are made up of 3 to 10 sugar molecules. 3 to 10 sugar molecules. And examples are ramenose, then stacose, then raffinose. The examples of oligosaccharides. Now talking about the polysaccharides, which we are going to talk about in detail. So polysaccharides. Polysaccharides. So the polysaccharides, they are of two types: homopolysaccharides and heteropolysaccharides. Homopolysaccharides and heteropolysaccharides. So the term here itself, homo, means when the same sugar units are there, it is homopolysaccharides. And if there are different monomeric units in the molecule, then they are called as heteropolysaccharides. So we will start polysaccharides from the simple example such as starch. Examples of polysaccharides. So first of all, we will be talking about starch. Starch. So starch is the plant storage form of carbohydrates. Plant storage form of carb. Then it is made up of amylose and amylopectin. Amylose and amylopectin. So amylose and amylopectin, and it is made up of what? This amylose and amylopectin, what are they? They are actually the chains of glucose only. So it is made up of glucose. Made up of glucose only. And they are linked with each other with the help of alpha 1,4 linkage for the linear molecule, and they are joined with each other. These glucose molecules, they are joined with alpha 1,6 linkage in the branched molecule. Branched. Just a second. For branched. Amylopectin is the branched form and amylose is the linear form. So the linear molecules are joined by alpha 1,4 glycosidic linkage, and the branched one, they are joined by alpha 1,6 linkage, and it is found in amylopectin. Right? Now moving towards the second example, that is glycogen. So this is the animal storage form of carbohydrate. Animal storage form of carbohydrate. And it is more dense than starch. More dense than starch. It's a branched molecule having branches, not a linear one. Then cellulose. So cellulose, it is connected with each other with the help of beta 1,4 glycosidic linkage. Beta 1,4 glycosidic linkage. And there is no cellulase in human. No cellulase in humans. Humans, they do not contain cellulase at all. And cellulose, they contribute in the fibers and they increase the bulk of the stool. Then dextrin. So dextrin is the partial degradation form of starch, or partial degradation of starch gives it. Partial degradation of starch gives it dextrin. Then dextran. This dextran is the plasma expander. It's the plasma expander. Then chitin. Chitin, it is present in the exoskeleton of insects. Present in exoskeleton of insects. Then lastly, inulin. So inulin, it is a polymer of fructose. It's a polymer of fructose. And it is used for the measurement of GFR, that is glomerular filtration rate. Inulin is a polymer of fructose and it is used for the measurement of glomerular filtration rate. because it is neither secreted nor absorbed by the renal tubules. Thus, it is used for the estimation of GFR, and it is best to measure GFR with the help of inulin because it cannot be secreted or absorbed in the renal tubules. Thus, it is the polymer of fructose. Now moving ahead and talking about the heteropolysaccharides. Heteropolysaccharides. So heteropolysaccharides, they are also called as glycosaminoglycan. Glycosaminoglycan. And it is also called as mucopolysaccharides. Mucopolysaccharides. So the two names, glycosaminoglycans and mucopolysaccharides. So when the constituents of the molecule is different than or is more than one type, if more than one monomeric unit is forming something, two types of monomeric unit is forming something, then it falls in the category of heteropolysaccharides. So the different sugar the sugar units are different over here. So constituent sugar units are different for heteropolysaccharides. So let us just understand the names. It's agar, then agarose, then hyaluronic acid, then heparin, then chondroitin sulfate, then keratin sulfate, then dermatin sulfate. So these are some of the examples of heteropolysaccharides, right? And why are they called as mucopolysaccharides? The term muco is something like mucilaginous. So they have got negative charge and they repel each other. And because of this negative charge which repel each other, it becomes slippery in nature. And that's why the name is mucopolysaccharides. And they act as very good shock absorbent, and they have this shock absorbing mechanism in the body as well, right? So this was about heteropolysaccharides, and now we will be talking about the important functions of carbohydrates. Important functions of carbohydrates. So first one is glucose. So it is the main source of energy. It gives 4 kilocalorie per gram. Main source of energy. Then it is important source of energy for RBC and brain. Important source of energy for RBC and brain. Then second is lactose. And lactose is the milk sugar, and it provides energy to the young developing babies. It's the milk sugar. Then the third one is sucrose. So sucrose is the cane sugar or table sugar. Then fourth is starch, and starch is the storage form of carb in plants. So it is storage carb in plants. Then next is glycogen. It's glycogen. So it is the storage form of carb in animals. Storage carb in animals. Then next is hyaluronic acid. And it acts as the shock absorbent in knees and in vitreous humor of eye and in the synovial fluid. So acts as the shock absorbent in the body at the various places, eyes, knees, then it is in the synovial fluid. Then next is heparin. This heparin is the anticoagulant and it is present in blood. It is the anticoagulant and it is present in blood. This heparin will stop the blood from clotting. Then next example is chondroitin sulfate. Chondroitin sulfate, and it is present in tendon, it is present in cartilage and present in bone. Chondroitin sulfate. Then keratin sulfate. Keratin sulfate, it is present in cornea. Then dermatin sulfate. Dermatin sulfate, it is present in skin, present in heart walls, present in blood vessels. Dermatin sulfate. Then next is dextran. And this dextran acts as plasma expander. And it is used in the treatment of shock. Then next is glycoprotein and glycolipid. So they are the important constituents of plasma membrane. Important constituent of plasma membrane. Then lastly, acts as the exoskeleton or can be used for the formation of exoskeleton in insects and cell wall of microorganisms. Right? So we have seen the functions of carbohydrates, right? So this was about the functions of carbohydrates. Now let us move further. What happened? Yeah. So now we'll be talking about heteropolysaccharides. Heteropolysaccharides. Heteropolysaccharides and heteropolysaccharides are also called as GAG. We have just seen when we were doing the examples of the polysaccharides. So the full form of GAG is glycosaminoglycan. It's glycosaminoglycan. GAG, glycosaminoglycan. Then it is also called as mucopolysaccharides. Mucopolysaccharides, right? So there are four important examples of heteropolysaccharides. The four most important examples. Four examples of heteropolysaccharides. What are the important examples over here? First is GAG, which is used for the the first one is agar. This agar, it is used for the culture medium, the media, the petri plates that we see. It is mostly made up of agar. So it is used for culture, used for microbial culture, agar. Then second example is agarose. So where have you heard this agarose? Have you heard it before? Yes, so many times we have heard about agarose. Agarose is used for electrophoresis. It is used for electrophoresis. So for the separation of DNA, for the separation of RNA, for the separation of protein, the slab which we make, in which there is these biomolecules, they run on the basis of their size and shape. It is made up of agarose. So that is also a heteropolysaccharide, agar and agarose. So they both are kind of gelatinous in nature, right? So what is their units, or what are they made up of? So agar and agarose, they both are made up of glucose and galactose. They are made up of glucose plus galactose. Made up of glucose as well as galactose, right? Then next is pectin. And this pectin is the dietary fiber. And the last in the heteropolysaccharide is GAG, which is the most common one. Right? Now talking about mucopolysaccharides. Mucopolysaccharides. Why the word given is mucopolysaccharide? Why the word given is so? So looking deep into the name, that it is called as mucopolysaccharide because the name given is because it was first time isolated from mucin. The name is given because it was isolated from mucin. So that's why the name given is mucopolysaccharide. And this GAG, it contains these disaccharide repeating unit. Very important. Contains disaccharide repeating unit. So the disaccharide repeating unit in GAG is made up of amino sugar and uronic acid. Remember this? It is made up of amino sugar and uronic acid. It is made up of amino sugar and uronic acid, right? So GAG, it contains the disaccharides, and the repeating units are amino sugar and uronic acid. So they both are present in combination, like they are the repeating units. So amino sugar plus uronic acid. So it should be written like this. This is the repeating unit in GAG. Fine. Okay. Now the repeating unit is this. So amino sugar. Talking about the amino sugar. So is the same amino sugar present in all, or the different amino sugars are present? Talking about the amino sugar. So amino sugars which are present over here, they can be of two types. That is N-acetyl glucosamine, N-acetyl glucosamine, or N-acetyl galactosamine. N-acetyl glucosamine or N-acetyl galactosamine. We'll be also seeing further that where they are present, in which molecule they are present and all that also will be seen. Now talking about uronic acid. Uronic acid. So this uronic acid is itself actually of two types, that is D-glucuronic acid, D-glucuronic acid, and the second type is L-iuronic acid, L-iuronic acid. And this L-iuronic acid is actually an epimer of glucuronic acid. So this is kind of same epimer of glucuronic acid, but they exist differently. So D-glucuronic acid or L-iuronic acid. Fine. Let us move further and let us talk about the properties of mucopolysaccharides. So properties of mucopolysaccharides. Properties of mucopolysaccharides. So the first property is it contains negative charge. Contains negative charge. So negative charge like what? Carboxylic group or sulfate group or acetyl group. And because of these groups, it contains negative charge. And this negative charge repel each other. So negative charge repel each other. And because of this repulsion in the negative charge, it serves two important functions. Because of this repulsion, there are two important functions. The first one is slippery action of hyaluronic acid. Slippery action of hyaluronic acid and mucus. And second, it acts as the shock absorbent. Acts as shock absorbent. Right? So slippery action of hyaluronic acid in the mucus and acts as the shock absorbent. Now talking more about mucopolysaccharide, in the same range of behavior, this repulsion with each other, mucopolysaccharide, what it does is it absorbs water, and if it absorbs water, it occupies greater space. And when it is squeezed out, it occupies lesser space and gets dehydrated. So on squeezing, water is removed, occupies lesser space, and gets dehydrated. So this action of mucopolysaccharide can be compared with sponge, or it can be compared with spring. So like a spring, it can be compressed, it can be leaved out, it will absorb water, it will behave like a sponge. So this all is falling under the category of negative charge. Because of that, all these things are happening. Now coming to the second thing, It occupies space in the extracellular matrix. Occupies space in extracellular matrix and acts as the ground substance in the extracellular matrix. Acts as ground substance. Ground substance in extracellular matrix, right? So the two properties, the negative one and the positive one, right? Now talking about the important GAGs or the important glycosaminoglycans. Important glycosaminoglycans. So first one is hyaluronic acid. Second is chondroitin sulfate. Then third is germatin sulfate. Fourth is heparin sulfate. Fifth is heparin. And sixth is keratin sulfate. So these are some of the important GAGs. Now talking about the composition of GAGs. So all the GAGs, they contain uronic acid. So now remember all these things what I am saying, that all GAGs, all glycosaminoglycan contain uronic acid. Except keratin sulfate. And this keratin sulfate, what does it contain if it doesn't contain uronic acid? So this contains amino sugars. It contains amino sugar plus galactose. And so remember, no uronic acid in keratin sulfate. No uronic acid in keratin sulfate. One thing. The second thing that you should remember is dextran sulfate. It contains amino sugar plus L-hyaluronic acid. Dextran sulfate. So it is UA plus AS in GAG, and for uronic acid, it's for keratin sulfate, amino sugar plus galactose. For dextran sulfate, it's amino sugar plus L-hyaluronic acid. Now, the third thing that you should remember is this amino sugar. Amino sugar is N-acetyl glucosamine in all except amino sugar is N-acetyl glucosamine. It's N-acetyl glucosamine in all except what? Chondroitin sulfate and dextran sulfate, right? And they contain, what does they contain? They contain N-acetyl galactosamine. N-acetyl galactosamine. They contain N-acetyl galactosamine, right? So we have done all the basics of heteropolysaccharides. Now talking about the examples of heteropolysaccharides. We'll be dealing with individual examples of heteropolysaccharides. So let us move further. The first thing that we are going to deal is hyaluronic acid. The first thing that we are going to see is hyaluronic acid. Hyaluronic acid. So it is made up of N-acetyl glucosamine, N-acetyl glucosamine plus D-glucuronic acid. D-glucuronic acid is made up of. Now where is it present? Present in synovial fluid, and vitreous humor. Then what are its functions? So it acts as shock absorbent, then it acts as lubricant, then it is responsible for cell migration, then it is also responsible for wound repair. So acts as shock absorbent, acts as lubricant, responsible for cell migration and responsible for wound repair. So this is about your hyaluronic acid. The students which are presently listening this video live, they can write in the chat box whether they are understanding the concepts which I am trying to tell them. Second is chondroitin sulfate. So it is made up of N-acetyl galactosamine plus D-glucuronic acid. Then it is present in where is it present? It is present in cartilage, bones and tendons. Present in cartilage, bone and tendons. Then its function. What are the functions of chondroitin sulfate? So it maintains the shape and structure. Now the third is dermatin sulfate. So it is made up of N-acetyl galactosamine, plus L-hyaluronic acid. right? Then it is present in, this is made up of N-acetyl galactosamine plus hyaluronic acid. Then it is present in, so as the word itself is telling, dermatan sulfate, so derma. So the most important place where it is present is skin. It is present in walls. Then what are the functions? So the functions are, it maintains the shape and structure, maintains shape and structure. Then it is responsible for, very important for MCQ point of view, that it is responsible for plaque formation and atherosclerosis. Responsible for plaque formation and atherosclerosis. Right? So this is about dermatan sulfate, that it is made up of N-acetyl galactosamine and hyaluronic acid. It is present in skin, in walls. The function is to maintain the shape and structure, and it is responsible for plaque formation in atherosclerosis. Now moving towards the fourth example, that is heparin. It is heparin. So the function of heparin is, first of all, made up of it is made up of glucosamine plus glucuronic acid. Then it is present in lungs, then mast cells, then blood, skin, etc. Then coming to the function of heparin, so if it is present in mast cells, it means it is present intracellularly as well. So talking about the functions, it acts as anticoagulant. Then it is present in capillaries, and it is responsible for, and lipoprotein lipase is released due to heparin, so it is responsible for release of lipoprotein lipase, responsible for release of lipoprotein lipase, right? So acts as anticoagulant, present in capillaries, and responsible for release of lipoprotein lipase. So this is about heparin, the fourth example. Now moving towards the fifth one, that is heparin sulfate. The fifth one, heparin sulfate. So it is made up of glucosamine plus D-glucuronic acid. It is made up of glucosamine plus D-glucuronic acid. Then it is present in Bowman's capsule of glomerular or glomerulus and because of the negative charge, it is responsible for selective permeability. Of the membrane of glomerular complex, and it will also act as receptor. So the function is that because of negative charge, it is present on the various cell surface. So this was about heparin sulfate, the fifth example. Now talking about the sixth one, that is keratin sulfate. So it is made up of N-acetylglucosamine, plus galactose. Right? And this keratin sulfate itself is of two types, that is keratin sulfate 1 and keratin sulfate 2. So keratin sulfate 1, it is present in cornea, and it is responsible for transparency of cornea. K2, keratin sulfate 2, it is present in cartilage. Right? And lastly, you should know two terms, that is proteoglycans and glycoproteins. So in both of the terms you can see glyc word, right? So proteoglycans, here also you can see the carb unit, here also you can see glycoprotein. So glycans and glyco, what is the difference? Why in one the glycans is coming in the end? Because proteoglycans, they are also made up of protein and carbohydrates, and glycoprotein, it is also made up of carbohydrate and protein. So why there is this shuffling of the terms? In proteoglycans, this glycan or the carbohydrate part, it is more than 95%, and here in glycoprotein the carbohydrate part is less than 5%. So this is majorly carbs, proteoglycans. This just there is a addition of protein molecules or small peptides in proteoglycans, and in glycoproteins the carbohydrate molecules are just present in a very small amount. So glycoprotein, so these are basically proteins. So the word which is coming in the end, that is the main thing, and the prefix is just the small part, right? So like this comes the end of the lecture, that we have completed carbohydrates in utmost detail, and this is it. I hope you people have enjoyed learning, you have gained something, and the concepts are quite clear, and you will be able to attempt the university examination, theory examination, as well as your PYQs, MCQs, and the competitive examinations, that is NEET PG. So thank you very much for your patient listening. Lastly, as we say, let's crack NEET PG together. Please subscribe our channel, that is Let's Crack NEET PG. Also hit the bell icon for getting all the notifications of the upcoming videos on our channel, and please like my video if you really enjoyed watching it and you have gained something from the matter. Then lastly, do not forget at all to use my referral code, that is FB01, for your class subscription. By using my referral code FB01, you will be getting a 10% discount in your plus subscription. So do not forget to use my referral code. Remember, it is FB01. So this is it for today's class. A very goodbye. We'll be meeting again with some other interesting topic of biochemistry. So this is it. Have a very good day. And